4 times. In 4 billion years. Only 4 times has a free-living organism crossed an invisible threshold and become something else entirely, something that rewrites the rules. The first time it happened, it created you and me and every complex form of life on Earth. The 4th time, it happened 100 million years ago. But we only proved it in the spring of 2024. This is the story of evolution happening in slow motion. A 26-year detective story. A mystery that began with a fragment of DNA floating in Pacific waters and ended with a discovery that could rewrite biology textbooks and change how we feed the world. They call it the Nitroplast, the newest organelle in the history of life. An organelle is a structure inside your cells, your mitochondria, your chloroplasts if you're a plant. These aren't just parts of cells, they used to be bacteria, free, independent, alive on their own. Then, billions of years ago, they were swallowed, and instead of being destroyed, they became essential. This is endosymbiosis. The word means living together inside, but it's more than coexistence. It's transformation. It's two becoming one. 1998. Jonathan Zare was hunting for something invisible. A marine scientist at the University of California, Santa Cruz, combing the Pacific Ocean for nitrogen-fixing bacteria. Nitrogen fixation. The ability to pull nitrogen gas straight from the air and convert it into ammonia, into life. It's a superpower. And for billions of years, only bacteria could do it. Zare was collecting seawater samples, filtering them, extracting DNA, looking for a specific gene, NIFH, the nitrogenase gene, the molecular signature of nitrogen fixation. And he found something odd, a DNA sequence that didn't match anything known, something widespread, something abundant. But when he tried to grow it in a lab, nothing. No colonies, no cells, no organism he could see. Just a ghost in the data. He called it UCYN-A, Unicellular Cyanobacterial Group A, a placeholder name for something he couldn't touch, couldn't culture, couldn't understand. On the other side of the Pacific Ocean, a paleontologist named Kyoko Hagino was facing her own frustration. She studied ancient ocean sediments at Kochi University in Japan, fossils of microscopic algae. And one species kept appearing in her samples, Braarudosphaera bigelowii, a beautiful single-celled alga, shaped like a 12-sided dodecahedron, covered in calcareous plates with 5-fold symmetry. Hagino wanted to study it alive, but B. bigelowii refused to cooperate. Every attempt to culture it failed, month after month, year after year. The cells would survive for days, sometimes weeks, then die. What Zare and Hagino didn't know in 1998 was that they were hunting the same mystery from different directions. And it would take another 14 years before anyone realized it. To understand what they discovered, we need to understand what an organelle actually is. Your cells are cities, bustling, complex, organized. And like any city, they have specialized districts. The nucleus is the command center. The endoplasmic reticulum is the factory floor. But two organelles are different: the mitochondria and the chloroplasts. They have their own DNA, their own ribosomes. They divide independently. Why? Because they weren't always part of your cells. More than 1 billion years ago, they were bacteria, free-living, self-sufficient, until an ancient cell swallowed them and didn't digest them. The bacteria kept doing what they did best. The swallowed bacterium that became mitochondria provided energy. The one that became chloroplasts captured sunlight. And over millions of years, something remarkable happened. Their genomes shrank. They lost genes. They became dependent. Not parasites, not just symbionts. Organelles. Permanent. Inherited. Essential. But here's the thing. We thought this only happened twice. Once for mitochondria, giving rise to all complex life. Once for chloroplasts, giving plants the power to feed the world. A third time we found evidence for it. The chromatophore in a strange amoeba called Paulinella. 3 times in 4 billion years. 3 examples, 3 textbook cases. Until April 2024, when Tyler Cole analyzed a proteome and realized he was looking at number 4. 2008. 10 years after Zair's first discovery. Still, no one had cultured UCYN-A. Still, no one had seen it. But technology had advanced. Massively parallel sequencing, the ability to read DNA from thousands of cells at once, even if you couldn't grow them. Zair's team collected water from Station Aloha, a research site in the Pacific. They used flow cytometry to sort cells, isolating roughly 5,000 UCYN-A cells from the ocean's soup, extracted their DNA, amplified it, sequenced it. And what they found was shocking. UCYN-A had the smallest genome of any known photosynthetic organism. 1.44 million base pairs, 1,200 genes. Most bacteria have several thousand genes. UCYN-A had the minimum. And it was missing entire pathways. No genes for making certain amino acids. No complete pathway for building nucleotides, the letters of DNA. No Rubisco gene, the enzyme that fixes carbon dioxide. It couldn't photosynthesize normally. It lacked photosystem II, the part that splits water and produces oxygen. How could it survive? Jonathan Zair remembered thinking, this genome looks like an endosymbiont. Something living inside something else, getting what it needs from a host. But what host? The data suggested UCYN-A was free-living, found alone in water samples. No obvious partner. It was a paradox. A genome that said symbiont, field data that said independent. 2012. 14 years after the mystery began, Kyoko Hagino finally succeeded. After more than 300 sampling expeditions across Japanese coastal waters, after countless failed attempts, she cracked the code for culturing Braarudosphaera bigelowii. The trick was patience, gentle handling, specific light cycles, trace nutrients in precise ratios. And when the cultures finally thrived, her microscope revealed something unexpected. Small spherical bodies inside the algal cells, perfectly round, 1 or 2 per cell. She didn't know what they were, but she suspected they were important. Meanwhile, 7,000 miles away, Anne Thompson in Zare's lab was analyzing DNA from ocean samples, and she found UCYN-A always appeared with specific haptophyte algae. Always together, never apart. She published a paper in Science, titled Unicellular Cyanobacterium Symbiotic with a Single-Celled Eukaryotic Alga. The alga was Braarudosphaera bigelowii, the same species Hagino had been culturing. The mystery DNA from 1998 and the mysterious spheres inside the algae were the same thing. UCYN-A lived inside B. bigelowii. But the question remained, was it a symbiont or something more? To understand why this question mattered, we need to travel back. Way back. 1.5 billion years ago, Earth's oceans were full of bacteria. No animals, no plants, no fungi, just single cells, swimming, dividing, surviving. And then something extraordinary happened. A large cell swallowed a smaller one, probably trying to eat it, but the small cell wasn't digested. It kept living, and it did something valuable. It breathed oxygen. It produced energy. ATP, the universal fuel of life. The large cell discovered it now had a power plant inside, and the small cell discovered it had protection, a safe home, nutrients delivered directly. Over millions of years, they became inseparable. The small cell lost genes it didn't need anymore. The large cell started controlling when it divided. This was the birth of the mitochondrion, and with it, the birth of all complex life. Your cells, my cells, every animal, every fungus, every plant. We all carry the descendants of that ancient bacterium, still breathing, still making ATP, still essential. Then it happened again, 600 million years later. A different cell swallowed a different bacterium. This one could photosynthesize, turn sunlight into sugar. And it became the chloroplast, the organelle that gave us plants, trees, grass, the green blanket covering Earth. Two events, two transformations, two times that endosymbiosis crossed the line and became something permanent. Scientists debated whether it could happen again. Some said yes. Some said these were unique events, one-time miracles. And then, in 1997, researchers found Paulinella chromatophora, a strange amoeba with what looked like tiny cyanobacteria inside. But DNA analysis revealed these weren't symbionts. They were organelles, chromatophores. A third example, a third crossing of the threshold. Much younger than mitochondria or chloroplasts, only 90 to 120 million years old. Still, 3 examples in 4 billion years, 3 data points. Not enough to call it common, but maybe not as impossible as we thought. 2013 to 2020. The years of intensive study. Zare's lab, Hagino's cultures, collaborators around the world, everyone trying to answer the same question. Is UCYN-A an endosymbiont or an organelle? What's the difference? Endosymbionts can leave. They can be separated from their hosts and survive independently. Legume plants have endosymbiotic bacteria in their roots, nitrogen-fixing rhizobia. You can extract them, culture them, study them alone. Organelles cannot leave. They are integrated. Their life cycle is controlled by the host. They import proteins made by host genes. They divide when the host divides. To prove UCYN-A was an organelle, they needed two pieces of evidence. 1. Protein import. Show that UCYN-A uses proteins encoded by bigelowii nuclear DNA. 2. Synchronized division. Show that UCYN-A replicates in lockstep with the host cell, that its division is controlled, not independent. Kendra Turk-Kubo, an assistant professor in Zair's lab, later said, "We started talking about this organism as a potential organelle many years ago, but we knew that we had a very steep hill to climb to be able to prove that to anybody." March 2024. The first paper dropped, published in Cell. Zair and colleagues from MIT, Barcelona, and Rhode Island. They measured UCYN-A and its algal hosts across multiple species, different strains, different oceans, and found something remarkable. The size ratio was constant. When the host cell was larger, UCYN-A was proportionally larger. When the host was small, UCYN-A was smaller. Perfect scaling. That's exactly what happens with organelles, Zair said. If you look at the mitochondria and the chloroplast, it's the same thing. They scale with the cell. They built a mathematical model, showing that the growth rates of host and symbiont were linked, controlled by nutrient exchange, metabolically coupled. They called it organelle-like, but they still needed more proof. April 2020. The world shut down. Labs closed. Experiments halted. But Hagino's cultures in Japan were thriving, and Zair's team needed those cells. Desperately. The plan was to use soft X-ray tomography, a technique that could image entire cells in 3 dimensions, see their internal structure, watch them divide in real time. The machine was at Lawrence Berkeley National Laboratory in California, run by Carolyn Larabelle and Valentina Loconti. But with pandemic restrictions, shipping biological samples across borders was nearly impossible. So they improvised. Valentina Loconti would meet collaborators in parking lots, both sides masked, windows cracked, passing precious vials of cultured algae like they were smuggling secrets. "We did what we had to do," Loconti later recalled. The samples made it to Berkeley, and the X-ray tomography began. Soft X-rays can penetrate water-rich cells. They reveal density differences, proteins, DNA, membranes, and because different structures absorb X-rays differently, you can see internal architecture. Loconti captured bigelowii cells at different stages, resting, preparing to divide, mid-division, and there it was, clear as day. The Nitroplast, UCYN-A, divided in perfect synchronization. When the nucleus divided, the nitroplast divided. When the mitochondria divided, the nitroplast divided. When the chloroplasts divided, the nitroplast divided. Frame by frame, cell by cell, the same pattern repeated. This wasn't random. This wasn't a symbiont doing its own thing. This was control. The host cell was telling UCYN-A when to divide. Exactly like mitochondria, exactly like chloroplasts. But there was still one more piece missing: the protein import. Tyler Cole was a postdoctoral researcher in Zair's lab. Young, driven, meticulous. His job: analyze the proteome, the complete set of proteins made by both B. bigelowii and UCYN-A. Remember, UCYN-A's genome is missing key genes. No complete pathways for amino acids. No full machinery for building nucleotides. If it's an organelle, those missing proteins must come from somewhere. The host nucleus. Cole isolated UCYN-A from the algal cells. Painstaking work. He needed pure samples, no contamination. Then he used mass spectrometry, identifying every protein present, comparing UCYN-A proteins to B. bigelowii proteins. The results stunned him. Over 350 proteins in UCYN-A were encoded by the algal nucleus, not by UCYN-A's own DNA. The host was making these proteins and sending them into UCYN-A. How? Every imported protein had a special sequence, an amino acid tag at one end. They called it the UTP, the UCYN-A transit peptide. Like an address label. Deliver to Nitroplast. Chloroplasts have the same system. Mitochondria have the same system. These transit peptides are the postal service of the cell. Proteins made in the cytoplasm get tagged. Molecular machinery reads the tag and delivers the protein to the right organelle. Cole found that roughly half of the proteins UCYN-A needs are imported. Essential enzymes for nitrogen fixation, proteins for amino acid synthesis, components for building nucleotides. It's kind of like this magical jigsaw puzzle, Zier said, that actually fits together and works. UCYN-A had thrown away genes, and B. bigelowii filled in the gaps. One genome, two organisms, seamlessly integrated. It is behaving as an organelle, Cole said. April 12, 2024. The paper published in Science. Title: Nitrogen-fixing organelle in a marine alga. Lead authors: Tyler Cole, Jonathan Zaire, Valentina LeConte, Carolyn Larabelle. 26 years of work, hundreds of ocean samples, decades of culturing attempts, thousands of cells sorted, parking lot rendezvous during a pandemic. And the verdict was clear. UCYN-A is not an endosymbiont. It is the nitroplast, the first nitrogen-fixing organelle ever discovered, the fourth primary endosymbiotic organelle in the history of life. The scientific community erupted. "This is solid science based on a lot of controls," said Verena Kreischbaumer, a plant cell biologist at Oxford Brookes University. Ramon Massana wrote in A Science Perspective, "The nitroplast represents a textbook case of a eukaryotic organelle. Ecology is the theater where evolution takes place." Tyler Cole, reflecting on the discovery, "The first time it happened, it gave rise to all complex life. Everything more complicated than a bacterial cell owes its existence to that event." 4 times. Mitochondria, 1.5 billion years ago. Chloroplasts, 600 million years ago. Chromatophores, 90 to 120 million years ago. And now, the nitroplast, also about 100 million years old. We are witnessing organelle evolution in geological real time. But the story doesn't end with the discovery. It begins there, because this tiny organelle might hold the key to feeding the world. Nitrogen, element number 7 on the periodic table. Essential for every protein, every strand of DNA, every molecule of RNA. Without nitrogen, there is no life. The atmosphere is 78% nitrogen gas, trillions of tons floating above us, but it's locked. N2, two nitrogen atoms triple bonded together, one of the strongest bonds in chemistry. Plants can't break it, animals can't break it, fungi can't break it. Only certain bacteria possess the enzyme nitrogenase, the molecular scissors that cut the triple bond and transform nitrogen gas into ammonia. For most of Earth's history, biological nitrogen fixation was the bottleneck, the limit on how much life the planet could support. Then came Fritz Haber and Carl Bosch. 1909, Haber discovered how to synthesize ammonia industrially, high pressure, high temperature, iron catalyst. Combine nitrogen gas with hydrogen from natural gas. 1913, Bosch scaled it up, built the first ammonia synthesis plant. The Haber-Bosch process. It changed everything. Farmers could now fertilize their fields with synthetic nitrogen. Crop yields exploded. Corn production in the United States went from 1,500 kilograms per hectare in the 1930s to over 10,000 kilograms per hectare today. A 2008 study in Nature Geoscience estimated that without the Haber-Bosch process, about half the world's population wouldn't have enough food. Nearly 50% of the nitrogen in your body came from this process. You are, in a very real sense, made of synthetic fertilizer. But there's a cost. The Haber-Bosch process consumes 1 to 2% of global energy. It uses 3 to 5% of the world's natural gas supply. It produces 1 to 1.4% of all CO2 emissions. That's roughly 450 to 500 million metric tons of carbon dioxide per year. The equivalent of all the cars in Germany, France, and the UK combined, every year, just to make fertilizer. And that's before the fertilizer is even used. When applied to fields, much of it runs off, pollutes rivers, contaminates drinking water, creates dead zones in oceans where nothing can live, releases nitrous oxide, a greenhouse gas 300 times more potent than CO2. The Haber-Bosch process feeds the world, but it's also slowly poisoning it. What if there was another way? Scientists have been chasing the stream for decades. Plants that fix their own nitrogen. Crops that don't need fertilizer. Some plants already do this. Legumes, beans, peas, alfalfa. They have root nodules, swollen chambers where bacteria live. Rhizobia, nitrogen-fixing symbionts. The bacteria fix nitrogen. The plant provides sugars. It's a partnership. But the world's staple crops don't have this ability. Wheat, rice, corn, they need fertilizer. Researchers have tried engineering nitrogen-fixing bacteria into these plants, tried transferring nitrogenase genes directly, tried breeding new symbiotic relationships. All have failed. The problem is oxygen. Nitrogenase is destroyed by oxygen, and plants produce oxygen during photosynthesis. Rhizobia bacteria solve this by creating low oxygen environments in root nodules. But engineering this in leaves, in stems, in cells actively photosynthesizing? It seemed impossible. Until the nitroplast. Here's an organism that fixes nitrogen in a photosynthetic cell during the day, while the host is making oxygen. How does it do it? The nitroplast is surrounded by mitochondria. They cluster around it, feeding it ATP, energy. And the nitroplast has lost photosystem too. It doesn't produce oxygen itself. So it creates a microenvironment. Low oxygen, high energy, perfect for nitrogenase. This system might provide clues, Tyler Cole said, into how such an organelle could be engineered into crop plants. Imagine wheat that fertilizes itself, rice that pulls nitrogen from the air, corn that doesn't need synthetic ammonia. No Haber-Bosch emissions, no fertilizer runoff, no dead zones in the Gulf of Mexico. It's not science fiction. It's a blueprint, already perfected by evolution, waiting for us to understand it. But we're still at the beginning. We know the nitroplast exists. We know it works. What we don't know yet is how to replicate it. February 2025. The American Association for the Advancement of Science made an announcement. The AAAS Newcomb Cleveland Prize, the oldest award given by the AAAS, recognizes the most outstanding research paper published in science each year. The 2025 winner, nitrogen-fixing organelle in a marine alga, Tyler Cole, Jonathan Zare, and their team. $25,000. But more importantly, recognition. This discovery, the committee wrote, fundamentally expands our understanding of cell biology, provides insight into the evolution of organelles, a process that was pivotal in the evolution of complex life. Jonathan Zare, now in his 60s, spent half his career chasing this mystery, from a DNA sequence in seawater to an organelle in a cell. Kyoko Hagino, over 300 expeditions, 12 years to grow one stubborn alga. Tyler Cole, a young scientist who proved what his advisors suspected for decades. And Valentina Locante, who met collaborators in parking lots during a pandemic. Because science doesn't stop, even when the world does. Kendra Turk-Kubo and the team are still studying it. How exactly does protein import work? What signals control the synchronized division? Can we identify more organisms in the process of becoming organelles? Because here's the thing. If it happened 4 times, it might be happening right now, in other species, other oceans. Endosymbiosis might not be rare. We might just be bad at recognizing it. The nitroplast is found from the tropics to the Arctic. It's globally distributed. It fixes a significant amount of nitrogen in the ocean, and we only just proved it existed. What else is out there? What other partnerships are evolving right now? What other organelles are being born? "4 billion years. That's how long life has existed on Earth. Single cells dividing in ancient oceans. For 3 billion years, that was all there was. Then 1.5 billion years ago, a cell swallowed another cell. And instead of ending in digestion, it ended in transformation. The birth of complex life. Since then, mitochondria have powered every animal, every fungus, every plant. Billions of species, trillions of individuals, all carrying the descendants of that first merger. Chloroplasts gave us forests, grasslands, algae blooms, the oxygen you're breathing right now. These weren't planned events. They were accidents. Cells trying to survive, bacteria trying not to be eaten. And out of those accidents came cooperation, integration, unity. The Nitroplast reminds us that evolution never stopped. We think of evolution as something ancient: dinosaurs, trilobites, the Cambrian explosion. But it's happening now, in every ocean, every forest, every drop of pond water. The Nitroplast evolved 100 million years ago. That sounds ancient, but it's young. Mitochondria are 15 times older. If mitochondria are grandparents, the Nitroplast is a newborn. And we're watching it grow up, watching it lose genes, watching it integrate, watching it become indispensable. We are witnesses to something that happens once in a billion years. And we almost missed it, because for 26 years, we couldn't see it. All we had was a fragment of DNA, a ghost in the data. But persistence paid off. Cultures grew, microscopes revealed, sequences decoded, X-rays illuminated. And now we know. There is a fourth organelle, a nitrogen-fixing factory inside a single-celled alga. A partnership 100 million years in the making. And it might just help us save the world. The Pacific Ocean holds secrets. Trillions of cells, billions of partnerships, millions of years of evolution. Jonathan Zaire dipped a bottle into that ocean in 1998 and pulled out a mystery. 26 years later, we have an answer. But the answer raises new questions. What else is out there, waiting to be found? What other transformations are underway? The Nitroplast teaches us that life is not fixed, it's fluid. Boundaries between species are temporary. Independence is negotiable. Cooperation can become permanence. Symbiosis can become fusion. And out of that fusion comes something new. Something neither partner could be alone. Your mitochondria proved it 1.5 billion years ago. Your chloroplasts proved it 600 million years ago. And the Nitroplast is proving it right now. 4 times in 4 billion years, 4 crossings of an invisible threshold. 4 moments when life became more than the sum of its parts. And the 5th time? It's already happening. Somewhere. In an ocean we haven't sampled. In a cell we haven't seen. Evolution never stops. It just waits for us to catch up. The 4th organelle isn't the end of the story. It's the beginning of the next chapter. This is Cosmic Canvas, where the smallest stories carry the biggest consequences. Until next time, remember that life's greatest innovations came from learning to share.